Good morning, everybody. My name is Guillermo Garcia-Manero from the Department of Leukemia at the University of Texas MD Anderson Cancer Center. And I'm going to give you a brief summary and update about some of the top clinical presentations on MDS at the ASH meeting this year. Of course, this year, the ASH meeting was really disrupted by the COVID-19 pandemic and the fact that we couldn't be there in person. Despite that, the quality of the presentations was, like every year, outstanding. And there were some presentations that I thought were really interesting. So let's go one by one. The first one actually is a presentation by the Spanish group on MDS, led by Dr. Diaz-Campello, looking actually at early intervention in patients with lower risk MDS with a del(5q) alteration, where patients were randomized to early lenalidomide versus observation. Of course, those patients were transfusion independent. This was a very interesting study, in my opinion, probably one of the best at the meeting. So traditionally, we have not treated patients with lower risk MDS that are not transfusion dependent. So in practice, if you see a patient with del(5q) MDS and the counts are normal and there is no significant anemia, most of us most likely will not start lenalidomide until the patient becomes transfusion dependent. Here, these investigators actually look at the effect of therapy versus no therapy with lenalidomide in transfusion independent patients with del(5q) minus MDS. And the data that these investigators showed is actually quite striking, even if it's kind of like an interim analysis, in that the outcomes of patients that received lenalidomide earlier during the course of the disease was significantly better than those patients that received therapy later. So the rate of transfusion dependency, the frequency, the timing, type of response, they were all in favor of early use of lenalidomide. So we think this is an important study because basically it suggests that starting lenalidomide, although the investigators, I have to say, use a little bit lower dose schedule than the standard of care, is associated with excellent quality of life in terms of complications and better responses and longer time remaining transfusion independent than those patients that just received drug later. So this has very important implications in our practice for a small subgroup of patients with this chromosome 5 alteration. The next study that was of great importance was to see the follow-up of the azacitidine Embelletoclax trial. This data was presented last year at ASH in a very important presentation by Dr. Wei. And there was a follow-up, this time by Dr. Garcia from the Dana-Farber on this multicenter trial. What we're seeing from these studies is that the original data presented last year at ASH is maintained with this follow-up with significantly longer duration of follow-up in terms of the analysis. We see that the combination of azacitidine with venetoclax is associated with a very high complete remission rate and overall response rate. And most importantly, that the duration of these responses and survival, this is not a randomized trial, this is a phase 1/2 study, appears to be longer than what you will expect with single agent azacitidine. This data is very important because we also learned in the early phases of the trial, this was the phase 1 portion of this, what is the proper dose, the type of toxicity, and how to manage these patients. So together with the data from the BLA study that led to the approval of venetoclax in AML, I think that this data really fortifies this concept of the combination of azacitidine and venetoclax in patients with high risk myelodysplastic syndrome. And the audience should be aware that there is a very important randomized trial known as the Berona Trial, randomizing azacitidine with or without venetoclax for frontline high-risk MDS that is starting with an endpoint of proving an improvement in survival that I think is going to be really a fundamental study in our field. And we are eager to participate on that trial. And of course, a few years from now, I see some of the data from that study. So in summary, the data from the updated trial of AZA-VEN in high-risk MDS is still looking very positive. The follow-up study that I'm going to discuss was presented by Dr. Sequeres from the Cleveland Clinic, who was actually a sub-analysis of trials looking at the combination of azacitidine and pevonidostat. Pevonidostat is a NET inhibitor, and a number of studies presented at EHA last year, we saw actually that this is associated with an increase in response rate and potentially improvement in survival as opposed to azacitidine. in small randomized trials. This population tested here is a little bit heterogeneous in that it also includes the so-called oligoblastic AML, meaning patients that have 20 to 30% blast that today are not considered in general as part of the typical MDS study. The bottom line is that this analysis that Dr. Sequeres performed on this azacitidine pevonidostat trial suggests that the combination of this doublet is actually superior to single-agent azacitidine. And part of this is related to the excellent toxicity profile that this NET inhibitor pevonidostat appears to have, meaning that we don't see excess myelotoxicity or some other unwanted toxicities. So this is important because we expect next year, in a few weeks, or few months, the results of the so-called Panther Trial, that was the definitive randomized study of AZA plus minus pevonidostat, that could be one of the drugs maybe approved next year for patients with high-risk myelodysplastic syndrome. And then finally, I would like to touch on another important study. This was, again, a follow-up of the combination of azacitidine with a TIM3 inhibitor known as sabatolimab. So TIM3 is another immune checkpoint, and this is an interesting molecule because it appears that it's not only present in the T-cell compartment, like PD-1 or CTLA-4, but also in the leukemia stem cell. So it is possible that a drug that targets TIM3 could have direct anti-leukemia effect, but also modulate the immune system as these immune checkpoint inhibitors like nivolumab or ipilimumab do. So the data was originally presented last year by Dr. Borate, and this year this was updated by Dr. Brunner from the Mass General in Boston. And what we're seeing here are two issues. Similar to what I described with Embelletoclax and pevonidostat, we see that the combination of azacitidine and the TIM3 inhibitor sabatolimab appears to give a higher response rate than expected compared to single-agent azacitidine. And even if the follow-up is short, we're actually seeing quite significant durability of these responses. In addition, we also see that the toxicity profile of this immune checkpoint inhibitor is actually quite benign, as opposed to what we have seen with drugs like nivolumab or ipilimumab in trials here at MD Anderson that have significant activity, but also quite a bit of toxicity. So to summarize these presentations, what we learned at ASH is the following: that early intervention in del(5q) MDS with lenalidomide may be something that we really need to consider in our patients, as this may result in longer period of transfusion-free time. This, of course, will be very important for our patients. And it brings some other concepts for treatment in the earlier stages of MDS, hopefully not only in those patients with del(5q) MDS. And then we saw updates with a number of doublets with Embelletoclax, with TIM3 inhibitor sabatolimab, with pevonidostat, that appear to be significantly more active than single-agent azacitidine. And the audience is probably aware that most of these combinations, actually all, are being tested in registration randomized trials. So if this data goes in this direction, we may have multiple new combinations for our patients with high-risk MDS pretty soon. This year at ASH, though, we missed data on updates with APR-246. We missed data on the updates with magrolimab, but these were extensively presented at EHA this year, and of course at ASH last year. I think that in the next 12 to 24 months are going to be very active in terms of what happens to the therapy for patients with MDS. To finalize, just to bring a word of caution that we're still missing more effective therapies for patients with lower risk disease. This year, we got the approval of luspatercept, but we need more of these compounds. And for sure, we need better, more effective therapies for patients with HMA failure, high-risk MDS. And with that, I want to thank you for this opportunity and listening to this short video. Thank you very much.